Okay, so I kind of wish I had put my talk a little later so that you had a chance to hear Professor Roland Winter, who will be coming to us through Zoom after the coffee break. And I'm going to apologize in advance for infringing a little on the coffee break. But I want to talk to you now about how to do all that cool stuff that Lois showed you under high hydrostatic pressure. Okay, so just a reminder, we are at a synchrotron, right? And some of you will saw that up close this morning. I noticed as you were registering, the people who would be doing hands-on tomorrow. So I'm looking forward to that. We've everything is running, and we've got actually Jose's group right now is collecting data, and so I'm looking forward to that. I just wanted to pop up, just so you know, the folks who are involved. Chin-Chu Huang, who's in the audience in the back, and myself run the beamline. And we'll be working with every one of you personally who come to collect data. And we love showing people how to do it. So Robert Miller is a grad student, and Gabrielle is in the audience. Both of them are in the audience. Ben Meyer is a fantastic undergraduate who is just finishing his degree. I can't talk about his work today. And then we have close collaborators, Nozomi, Saul, and Kathy, whom we rely upon for a lot of advice and guidance. I also want to draw your attention to, we have a separate high pressure crystallography initiative, which is younger, but we are starting to do outside user samples successfully. And Zhongwu Wang, who I think is also in the audience, he was at the banquet last night, and Saul will mention that a little bit, but I think that you'll see more of that in the future. And then I want to point out Steve Meisberger, who is a brand new Mac Chess staff scientist who will be in charge of the line that does that. So just so you know the folks. Okay, so this is Richard's big picture of the stuff that happens in nature under pressure that you can see with SACs. And this is part of a methods in enzymology chapter. There's a nice big volume coming out, actually 2 volumes that John Tainer is editing, full of a wealth of how-to SAC stuff that will be published. The first volume is under review right now, and I have a section in it on the pressure stuff. So please look for that. Some of you have some early drafts. So there are sort of 3 broad categories. You can see the pressure axis, 0 megapascal, I call it atmospheric pressure, up to 100 megapascal, it's the bottom of the ocean, the Mariana Trench, 110 or so. Then 300 and 500 are sort of food science kinds of pressures, or if you go down in the deep subduction zones, just before it's too hot for life, maybe 120 some Celsius, you might have pressures in the 300 to 400 megapascal. So those are the pressures that we know about where life may occur. So molecular complexes is a broad category. I've sort of represented it very schematically here, as a dimer that dissociates and unfolds. But you could also, I think Roland will talk about DNA, RNA, nucleotide kind of things as well. So I meant that to be a very broad category of molecules individually. Lipid mesophases, another fascinating area. I'll show you an example. That's a really fun system to work with because they're strong scatterers and dramatic changes take place, and they're very relevant to life, right? Everybody's got a membrane that we know of. except the last category, right? So liquid-liquid phase separation is a hot new area. It's actually coacervates have been around for eons, but it is now recognized that living systems don't necessarily need membranes to concentrate things. And so you can see this thermodynamic separation of phases, especially under conditions with high salt and stuff like that. And I hope Roland in his talk will mention some of the very interesting pressure work that's going on, where you pressurize and a phase can go away and then a phase can reappear again. And SACs can tell you about that as well. So I'll get back to these a little bit later. So Lois did a beautiful job of, better than I can, of explaining the whole SACs, but I just want to remind you the scattering profile, right, is the piece of data that we collect at the synchrotron. It's a one-dimensional curve. You can kind of think of it as a sunrise, right, where the sun is just below the horizon. That's a direct X-ray beam, and you see all this lovely color and brightness fade into the darkness overhead and early in the morning, right? So that's small angle scattering, and it's that one-dimensional curve, okay? So Lois also showed you the components that make up the scattering profile, right? There were 3 components. I wanted a prefactor, which I'll talk about in a moment. And you use that to get molecular mass and some other useful information. The form factor, which is what most molecular biologists, structural biologists are interested in, because that tells you about the shape and the nature of the individual molecule in solution. You get radius of gyration, molecular shape, maximum dimension. You can tell folding, right, which is a very powerful thing. SACS does not require freezing or crystallization, right? It's a solution method. And it doesn't stand entirely on its own. That's why I've got an MR person here today, an EPR person, spectroscopy person, right? These solution techniques, need to work together, and the sum is greater than the whole. So, and then there's some wonderful atomistic modeling stuff that Lois talked about. I won't talk about any of that. And then over here, the structure factor, which for most structural biologists just work with dilute solutions, and that is equal to 1 and we ignore it. But actually there's some it's somewhat underutilized information. And it's a little beyond the scope of this talk to really talk about that, but I'll mention it a little bit later. And I think that Roland may mention that in his talk about liquid-liquid phase separation and osmolytes and stuff. So there are pressure effects in all of these things. But I wanted to just talk about some boring math for a moment in the prefactor, because as you apply the methods of standard SACS, it is important to recognize that you can't just casually do everything you used to do in ordinary SACS without thinking about, does pressure affect the underlying assumptions? As I read the history of SACS, some of these grand old textbooks, you go back to Guinea and so forth, you become impressed at the brilliant approximations that were done, which are very valid and very powerful and have been forgotten in the modern age because we use the techniques so casually. So we need to go back and think a little bit under pressure if these, so I just wanted to stop for a moment. So this structure factor thing, I'm very excited about, right? There's quite a community of people, and this has implications for desiccation of organisms, right? Where they're very concentrated. There's not just a little thin layer of water between the proteins, low water activity, crowding, the effect of osmolytes, right? I can't really get into that much here. There's not enough time. So sorry for the equation, but I just wanted to tell you about K, right? The prefactor. And the important term here is contrast, all right? That prefactor in SACS actually, and neutron scatterers are well aware of this, and some SACS groups like Lois's group are aware of this, that the scattering intensity depends upon the difference in average electron density between the molecule you're looking at and the solution and the buffer. And you can play with that contrast. Neutron people do elaborate deuteration chemistry to take advantage of differences in scattering contrast between nuclei. And you can also do those same kind of games with X-rays by changing additives like sucrose in your buffer, right? And Lois's group has done a lot of this also with RNA, DNA kind of stuff. So, so what's interesting is that in the customary approximation, I have here, the contrast is a difference between the rho of buffer, which is the density of water, and the density of protein without water, right? Which people approximate as the mass electrons per gram divided by the specific volume. And that's a number that, so that's a number which it is commonly believed, based on the measurements that have been done in the literature, doesn't change very much with pressure. That's an assumption, it's an observation, 10 times less variation. So we can assume for the purposes of this discussion, it's constant. It's not, of course. I'm not even sure exactly how to think about this number atomistically, but this is something that's measured from ultrasonics, velocity of sound and stuff like that. And so if you assume that water is more compressible than your protein, then what you find if you look at the ratio of intensity of your measurement, your Sachs measurement at ambient pressure to high pressure, it's a ratio that depends upon how much water has compressed. And you'll see that at 100 megapascal at the bottom of the ocean, you're already down to 80% intensity. The contrast has reduced. And by the time you get to 400 megapascal, where proteins are beginning to unfold and do really interesting things, you're talking about less than half of your intensity. Now, so it's important to remember that this has an impact on the things that you use, right? So here's an example of a Kratky plot, right? This is Q squared times I, right, as a function of that scattering angle. And you typically, for a folded protein, and this is glucose isomerase, which is a rock that we use as a standard. So it doesn't do any unfolding or anything at 300 megapascal, but you can see the intensity is going down. And that formula predicts it really well for this rock. So what are the implications for the standard stuff that you do with Sachs? So one of the things that you do is measure molecular weight, right? So that you're interested in, do I have a dimer, a monomer, a tetramer? Are they in equilibrium, right? So even though Sachs is not terrifically great at molecular weights, it's an important piece of information that you get. And the first way is we can calibrate, put our scattering on an absolute scale in units of inverse centimeter using a water standard, and you plug it in and you get the molecular weight. But guess what? It depends upon the contrast. And we know that contrast changes with pressure. So this method is biased. You could try correcting it if you think you know what that delta rho is, but we recommend against doing this. So don't look at that number unless you're prepared to think about contrast, all right? So, I thought I just mentioned this. You'll notice at the beamline when you collect data, here's typical raw data, that as you pressurize, the baseline goes down. And I was always very curious about why that was. And we have our data is normalized for transmitted intensity, so it has nothing to do with increasing density per se. But if you look at the scattering of pure water as a constant that depends upon the density and the compressibility, right? So neat liquids, you can think of them as fluctuating densities, and the amount of fluctuating density scatters X-rays, and that's what gives rise to scattering of a pure liquid. And if you plug in the numbers, what you will find is both temperature and pressure affect that baseline, and pressure actually affects it just as much as temperature, and they go down. So just so you know, when you're on the beamline, that's what's going on, I believe. So the other method of protein standards, which you may have obtaining molecular weight, is using a standard. Now, in principle, you could use a standard if you want, but you've got to be sure that it's at the same pressure that you're measuring, right? You could do that. It would be a lot of work. Do a glucose isomerase at all the pressures. Use a different standard for every pressure, interpolate. But just the same story. Concentration-dependent methods. Don't look at them unless you know what you're doing. Okay. So the third method, there are as a class of methods that do not depend on concentration to get molecular weight. They depend on these integral invariants. You see, it's an integral V. This one is the Perrault volume, which is an integral of the Kratky plot, basically. And you will notice that those K factors cancel. So this baby is contrast independent. So that makes it much more trustworthy. Now you're still, when you're calculating molecular weight, you still have to make an assumption about your protein specific volume. That's an underlying assumption that we don't think about. Then they use average values obtained by measuring a bunch of different proteins. So, but that's likely to be a small effect. So I recommend when you look at molecular weight, use the integral methods Don't use the other methods. And there are more sophisticated techniques. I haven't looked at them to see, so just be cautious. Okay, like, well, this one I probably trust too, the volume of correlation, right? Same kind of integral. Okay, now, this is, Lois talked about this, the classic Guinea approximation, right? You can look up his thesis, it's in the library right here. And that's where it's from. So he, yeah, we should, all students should aspire to come up with a formula like that, that you know, what has it been, 100 years now? Is as important as ever. So you get this line when you plot log versus q squared, but it is not totally, anyone who's done neutron scattering, right? I know Kushal is out there, and will know that there's this thing called the Sturman plot, right? A long time ago, people were looking at contrast, both X-rays and neutrons in the 1960s, and noticing that radius of gyration changed with contrast. And so, you know, the neutron folks, so this, what they've done is taken, this is the contrast, right? Average solute, average minus average solvent. And they assume that your protein is a solid with uniform density plus all of the deviations from constant. And they came up with this formula, and you make these plots, and you can sort of interpret from those sort of how dense, how lopsided the density is. So I don't know how important this is in high pressure. But if you're looking for small changes, which you frequently are, pressure effects are not always large. If you're looking at little changes, you need to be thinking that it might have something to do with this, especially if your contrast is getting small, right? And it does for X-rays. For neutrons, it goes the other way. Okay, so the take-home messages here is be careful when you're interpreting small changes due to pressure. Don't just casually use the classic normal tools. Be careful, some of them have contrast bias. And this is what I wanted Lois in the audience for. Somebody really needs to try a pressure-induced contrast variation experiment to see, because contrast variation is a powerful tool when it works, right? Kushal has done that too. Yeah. Is there enough contrast change in a pressure experiment that you have got a dial where you don't need to add anything, you don't need to deuterate anything, you just crank up the pressure? And can you learn, can you distinguish DNA from protein and so forth? So that's a challenge to everyone in the audience. You know, somebody needs to try that. Okay, all right. So I just want to, let me look at the time. Sorry. What time is it? Oh, here it is. All right, we're almost time for, good, all right. We're almost time for break. So this is the setup that you'll get to see, right? So it's a block of really tough metal with some diamond windows in it here, and we use water as a pressurizing medium. Your sample is a disposable cell. Chinchu and I will teach you how to load those cells, and you put it under pressure that way. And this is a very easy system to load and unload. I think people have found this, so we're able to be very productive and generate a lot of data. It's not that hard to do, okay? Still, you have to load them manually, all right? So I won't go into this. I'm sure Roland will talk about it, and possibly George later on. So proteins unfold, right? That you may have heard that. It's counterintuitive, and Professor Winter will give you a better understanding of why that happens, I think. But that's the first phenomenon. If you take it up above, you get above 300, you start to see things unfold. Why don't they get smaller? They unfold because that's how they reduce their volume, actually, because they're not perfectly packed. So what does an unfolded thing look like? I'm going to show you, there's several, we can actually, in addition to static sacks, you know, where you load a sample manually, put it in, we can do chromatographic high pressure sacks up to 100 megapascal, not as high a pressure. That's what this is. So we inject a sample. This is one of Kathy Royer's favorite things, PP32, which is a single small protein that's got cavity that happens to unfold at a nice low pressure of 100 megapascal. And you inject it and you see your chromatographic peak, and you can measure Sachs properties as the peak comes out, right? So the radius of gyration is the first one, and look what happens. The contrast goes down, and the radius of gyration goes up, right? It's unfolding, it's unfolding. And this is what, so the Kratky plot, now it's kind of subtle, right? The tail goes up as you become unfolded, right? And it's easier to see in this P of R thing, right? Which is the pair distance distribution function, which is kind of represents, you look at all the electron pairs in bin histogram, right? It's like, it's real space, that's R, and this is like a population. And you can see that the thing has gotten larger, okay? That's what unfolding looks like. A little bit subtle in this example, but that's what Sachs is good for, telling unfolding. I just wanted to note that because of the growing interest in intrinsically disordered proteins, that the Sachs community is beginning to develop tools to deal with them. And so if you go to the Sosnick group website, they have a wonderful server that helps analyze some of these disordered systems. So that if you go to high enough pressure, you've got some disorder, you've got unfolding, there are beginning to be tools for sort of analyzing those things, thinking about ensembles, right? Which these things are not a single species, they're ensembles, right? Okay. So I just want to talk about a few examples now. And those of you going to the Beamline will get a chance. We have prepared, we have both yeast enolase and well, I don't know if we have prepared yeast, but we could make some quickly. Lactate dehydrogenase, which are examples of oligomers that bust up when you put them under pressure, right? So this is pretty simple thermodynamics. It's an Arrhenius expression, right? So their atmospheric equilibrium constant depends upon volume change that it takes place. And the volume change is not just the volume of the protein itself, but the water surrounding it, right? You might have a cavity that may or may not have well-packed water molecules. You pull it apart, it's a little better packed. The volume actually went down. So that's, so volume, so pressure sensitive stuff, as I'm sure Roland will tell you all about, is all about volume changes. And volume changes are fascinating, right? Because important, physiologically important, because proteins are not just, well, they're mostly rocks, but they need cavities in order to function. They need flexibility in order to function. And these things are things that pressure can affect. Okay, so yeah, so there's lactate dehydrogenase. It has that enormous red void in it, right? It's probably why it dissociates. Okay, so, and I'll direct you, again, there's a wonderful review by Roland Winter here. I may even have some more recent references, but that's pretty recent. Yeah, okay, so we'll talk, now, can Sachs follow dissociation? Sure can, right? They used to do this with spectroscopy a lot, right? If you have a spectroscopic signature of dissociation, and this is a Fujisawa paper. It gets a very nice paper because it's about Sachs, right? And you can follow the radius of gyration as a complex busts up. And you could, in principle, although I don't think anyone has done this yet, with Sachs information, if you derive alpha, which is a measure of the degree of dissociation, you can fit this line as a function of pressure and get the KD and the volume change associated with that. So they do that a lot with spectroscopy, right? But you should be able to do it with Sachs. All right. So here's an example. This is yeast enolase, right? Which is a dimer, which at 4 degrees C under just 100 megapascal will break up, and you can fit, we know what the crystal structures are, and you can fit them to the data. And sure enough, at zero ambient pressure, it's a dimer, it's all dimer. And at 100 degrees, it's actually a mixture of 50-50, right? And I'll show you how you can do that. If you can tell what percentage, it's really easy. This is really easy stuff if you know structures, all right? So yeah, this is the technology that we're most excited about, is the ability to actually separate stuff under pressure. Chromatography connected to Sachs was an important revolution. It seemed like kind of an obvious thing, but it turns out to have been extremely important because aggregates and mixtures are trouble for Sachs, right? And this helps separate it. And so at least 50% of people using normal sacs are doing chromatography first. And I recommend, if it weren't for the workshop starting at the beginning of this run, I would recommend that everyone interested in going into pressure sacs try ordinary ambient sacs first so you understand your system, and then you're ready to do pressure, right? But we can do chromatography under pressure up to 100 megapascal and at temperatures 0 to 50 degrees C. So we can do temperature pressure stuff. Yeah, okay, here's lactate dehydrogenase. You can see that cavity again. So there's a program that you can use if you know the individual structures, right? The sacs mixture is a linear combination depending on the volume fractions. You can calculate the fit between the theory and the experiment and optimize it. And so, all right, so here's yeast enolase. This is a chromatogram, right? It's a single peak, nice clean sample. And you calculate, is it monomer, dimer, or a mix? See the blue, it's 100% dimer, right? Nothing interesting happening here, ambient pressure. If you pressurize it to 100 megapascals at 4 degrees C, look at what's going on. The curve, right, it's becoming a mixture. You can see, you can't separate this mixture very well. There might be a little bit of separation here, right? But, and this is known, yeast enolase is a reversible dissociation. You take the pressure off, it goes right back again, instantly, right? So the chromatograph can't really separate it efficiently, right? But some years ago, it was noticed that not everything does that. So this is lactate dehydrogenase. Sorry, this is a confusing slide. That is a single peak with a molecular weight. I don't have a pointer here, do I? Okay. Up. I think I can use my cursor. Oh, you're right. Yeah. Okay. Here. Yeah. So this green curve is the ambient pressure. It's single peak. Well, it's got a little aggregate there, which is why sacs is good. Why sacs is good. And look, the molecular weight is 100% tetramer, totally predictable. Then you put it under pressure, and look what's going on. There's a second peak has formed, and it's got lower molecular weights. And if you go through the process I just showed you, you can calculate that that second peak has got an awful lot of dimer. It broke from a tetramer to a dimer. It's not a perfect separation. And this is a case of the so-called, what they called back in the, 20 years ago, they called it, well, shoot, my memory just failed, right? So the idea is that if something dissociates, it might rearrange a little bit and have trouble going back together again, right? So some complexes do, some don't. And that's what you're seeing here. It's separable because when it comes apart, the kinetics are such that it doesn't go immediately back together again. So, yeah, all right. So we focused a lot on proteins because that's traditionally what we've done. And we have a big community of people who are very interested in that. But I want to talk just briefly about the other things here, right? So lipids, super important. And they can form different kinds of so-called mesophases, right? So this is the lamellar fluid is the one most important to biology. All of membranes, it's a fluid membrane that has fluidity. If you pressurize it, it becomes a rigid gel and it can't do its function, right? So a lot of life is believed to adapt the chemistry of these things to prevent this gelation from happening. But also, there are other interesting phases, and they are visible in sacs because they have repeats. Remember, Lois talked about repeats. You see these repeats and they're strong peaks. So the inverted hexagonal phase is instead you have the hydrophilic stuff all bunching together and the tails, hydrophobic tails splaying out and touching each other. And what do you notice about this structure versus this structure? Yeah, so this one doesn't look very efficiently packed, does it? Yeah, and so it's pressure sensitive. You pressurize it, busts apart. This may be physiologically important, right? When membranes have to do things, they have to touch and budding and stuff like that. They may have to go through these kinds of phases in order to do that. So We had a terrific collaboration that a group, Itai Budin, who's a lipidomics person at UCSD, Jacob Wittikoff, who is a graduate student working at the Monterey Bay Aquarium with Steve Haddock, and they have studied the lipids of ctenophores, which look a lot like jellies, but they tell me that evolutionarily they're as far from jellies as we are, but they look like jellies. So they harvest the lipids, and if you put those in the X-ray beam, you get these beautiful patterns. You don't even need to background subtract, right? So here's the lamellar phase. You can see the lamellar is these beautiful regular spacings, like Lois was talking about, regular spacings. And then the inverted hexagonal, look at that. Okay, so there are these beautiful, clear, these are pretty easy experiments. And these guys, this is a model system, but real systems are complicated mixes, but they are making headway and seem to be able to interpret the phase behavior of the real lipids of these systems and how they adapt. So that I think is very exciting, very relevant to deep life and things like that. So, and then once again, I just wanted to point out that and there's no time to talk about this. I hope that Roland covers it a little bit in his talk, that this exciting new area of liquid-liquid phase separation, concentrated materials, that it's the structure factor, right? Which is when you become concentrated, you're starting to get scattering between neighboring molecules. So it tells you about the organization and the arrangement of molecules. And actually, in the earliest days of X-ray scattering, people were interested in atomic fluids and simple molecular fluids. And so they weren't interested in the structure of a water molecule. Everyone knows what that looks like. They were interested in the arrangement. And so quite a bit before the days of computers and synchrotrons of theory went into trying to use X-rays to figure out the distributions of molecules in the fluids. So there's a wonderful whole area that's grown out of that, and it's related to colloid science as well, which uses this information. And I wish I could do a whole nother lecture in that. And we are beginning to gain experience in how to do these. If you like, I can prepare concentrated lysozyme and you can try this on the beamline. Okay, all right. So with that, I will finish and point you to this wonderful review that the RCN, Kathy's RCN, and all of the collaborators have written. And then coming soon, please look for my high pressure sacs, deep life and extreme biophysics methods and enzymology chapter, which has all of this stuff and more, and a big review and overview of everything. So thank you very much. Yeah.